Hello and welcome back to another video. It's been a while since you had one of these from me. In this video, we're going to talk about antibiotics. We've already introduced the concept of antibiotics and their targets before. Today, we're going to look very specifically at targets of individual antibiotics and the functions. So how these antibiotics work to kill bacterial cells while not killing our own cells. So first up, let's look at this question of where do they come from? The original antibiotics, the first ones that we discovered, came from molds. So you've heard of the drug penicillin. It comes from the Penicillium mold. There's another drug called cephalosporin that comes from the Cephalosporium mold. So these are naturally occurring antibiotics. Both penicillin and cephalosporin are what we call beta-lactam antibiotics, and we'll talk more about that on the next couple of slides. Most of our antibiotics that are naturally occurring are from a bacterial genus called the Streptomyces. So there's a variety of these species that can make antibiotics, and they make ones you may have heard of like tetracyclines. They make the aminoglycosides, macrolides, and a whole bunch of other ones that you may or may not have heard of. And then finally, bacteria belonging to the genus Bacillus. Again, a number of species can make these antibiotics. Most of them are what we call polypeptides. So they're small amino acid chains. And this is like bacitracin that we've talked about before, which is the active ingredient of Neosporin. Now, way back at the beginning of the semester, we hypothesized targets of antibiotics based on bacterial structures. And of course, we said, let's target the cell wall because humans don't have a cell wall. These cell wall synthesis inhibitors can target peptidoglycan synthesis and also target transpeptidation, which we'll go over in a few minutes. Ribosomes are another big target because recall bacteria have 70S ribosomes and we have 80S. Nucleic acid synthesis, usually by targeting DNA gyrase. There is one very important drug that targets a specific metabolic pathway of bacteria. These are called antimetabolites. And then finally, we can have specific antibiotics that target the gram-negative outer membrane. So maybe they target particular structures that are only found in our gram-negative cells. Let's start by talking about the cell wall. There are a number of antibiotics that target the cell wall. We're going to look at these four here really briefly. So beta-lactams, these would be things like our penicillins and variants of penicillin. Semi-synthetic penicillins means that we've taken regular old penicillin, modified it in the lab to make a semi-synthetic. So semi-synthetic because it's based on the naturally occurring penicillin and has been modified in the lab. Cephalosporins are also beta-lactams, but again, they're made by a different mold. Our polypeptides, like bacitracin, and then the glycopeptide vancomycin, which you may have heard of before. All right, penicillin. Penicillin is called a beta-lactam because of this red ring right here is called a beta-lactam ring. And this ring is really important in the functionality of the drug. Now, penicillin is bactericidal, meaning it will kill bacteria and not just inhibit their growth. And it targets peptidoglycan synthesis. We'll look at that in the next slide. One of the main problems with penicillin is it's what we call a narrow spectrum drug. That means it will only target a few select species or types of bacteria. And it's particularly good against our gram-positives like streptococci, staphylococci. And it is not very effective in its regular traditional form against gram-negative bacteria. So mostly against our gram-positive for regular plain old penicillin. This is how penicillin and semi-synthetic penicillins work. The gray structure here is called penicillin binding protein. It's the enzyme responsible for carrying out transpeptidation. It binds to our peptide cross bridges here, and it forms the bond that you can see between them. And again, this is crucial in maintaining peptidoglycan integrity so that the layers of NAG and NAM don't simply fall apart. Penicillin and its derivatives are shown in these sort of orange P or 9 shaped things here. And this actually binds into that pocket of penicillin binding protein where it would normally bind our peptide bridges. Okay, so where it would normally bind to these, penicillin fits right into that active site of the enzyme. Now that penicillin is in here, you can see it blocks penicillin binding protein from attaching to our amino acid chains because the active site is taken up, and no more cross bridges can be formed. Now, penicillin itself is the natural product, and again, our semi-synthetics are ones that are modified in the lab. These are some that you've probably taken before, like amoxicillin. It's given pretty regularly to children for things like ear infections, ampicillin, maybe even methicillin. That original mold product of penicillin still has its beta-lactam ring, but additional side chains have been added. So off of one side or the other of the base penicillin molecule, different structures have been added. And these structures tend to allow for a broader spectrum of the drug. So not only will it target gram-positives, but now it's actually effective against a variety of gram-negatives as well. Next up in our cell wall attacking drugs are the cephalosporins. These are made by the Cephalosporium mold, so again, a natural product. They do have a broader spectrum than naturally occurring penicillin does, which is really nice, and they also have a lower toxicity. Like penicillins, cephalosporins also have this beta-lactam ring, but they have some different structures. They have a side chain on each side rather than just the one side chain that we see with penicillin. But they act similarly, so they bind to penicillin binding protein and prevent transpeptidation, or the forming of those cross bridges between our NAG and NAM layers. Bacitracin and our polypeptides are the next on our list. These are generated by bacteria. They're generated by species of the genus Bacillus. They are, again, effective mostly against gram-positive bacteria, and it's the active ingredient in Neosporin. It is not a beta-lactam. The structure is shown over here on the left, and you can see it's pretty big compared to penicillin and cephalosporins. It also works differently. Recall that NAG and NAM are made in the cytoplasm of the cell. NAM is then bound to a UDP molecule, and the cellular protein bactoprenol binds the NAM, and this protein itself actually flips to put our NAM on the other side, the outside of the cell membrane, and then our peptide cross bridges can be formed. Bacitracin binds to bactoprenol and prevents the association of bactoprenol with NAM. So it prevents the translocation of NAG and NAM through the cell membrane so that they can form the cell wall layers. So it still prevents cell wall synthesis, just in a very different way from our cephalosporins and our penicillins. The last cell wall targeting antibiotic that we're going to talk about are the glycopeptides, and you may have heard of a specific one called vancomycin. Vancomycin, the structure shown here on the left, again, it's really big compared to penicillin and cephalosporin. It is a naturally occurring drug made by this bacterial species right here that I'm not even going to try to pronounce. The issue with vancomycin is that in most cases it has to be delivered IV. So we have to have a patient in the hospital to deliver this drug because we need to monitor them and make sure they don't have a bad reaction to vancomycin. And this is one of those drugs that we tend to reserve its usage. So we will use it only as a last resort because we don't want to drive resistance to this antibiotic. As antibiotic resistance increases, it's important that we keep some antibiotics that we know will still be effective, and vancomycin is one of those that we use only when nothing else has worked. The way vancomycin works is different from both our penicillins and cephalosporins and from bacitracin. In this instance, our vancomycin, which is the blue claw-shaped kind of things here, is going to bind to those amino acid chains. So the chains that would normally have cross bridges formed between them to link our layers of NAG and NAM, vancomycin binds to the end of those chains. With vancomycin bound to the ends of these amino acid chains, our penicillin binding protein here can't fit in there, can't bind those chains to form the cross bridge. So even though it works differently, it is still preventing transpeptidation. And of course, without transpeptidation occurring, our NAG and NAM layers will fall apart and our bacterial cells will die. The next really big group of antibiotics targets ribosomes and inhibits protein synthesis. And there's 4 big classes of these antibiotics, so the tetracyclines, chloramphenicol, aminoglycosides, and macrolides. These are not just individual antibiotics, but tetracyclines, aminoglycosides, and macrolides are classes. of antibiotics, and there are multiple drugs within those individual classes. So if we look based on just sheer numbers, there's probably more drugs that target the ribosome and protein synthesis than any other bacterial structure. There are 8 big types of tetracycline. They all share a similar structure that you can see over here, where we have multiple carbon rings linked together with various side chains coming off of them. One of the most important of this class is doxycycline, which is a semisynthetic. So it's a modified version of the original tetracycline compound. This is doxycycline shown at the bottom. It's important because it's used to treat some intracellular pathogens and some otherwise challenging to treat pathogens like Lyme disease. Naturally occurring tetracycline is again made by the genus Streptomyces, and it is very broad spectrum, so they can target gram-positive and gram-negative cells. The only drawback to tetracyclines is that they have been overused historically, and there is a lot of antibiotic resistance that is now built up to these drugs. So they're not always as effective for treatment as they would have been, say, 15, 20 years ago. Now, the way tetracyclines work is that if this is our little tetracycline molecule right here, they bind into the A site of our ribosome. And by binding there, they prevent our tRNA from entering the ribosome, effectively blocking translation from occurring. If you cannot get a new tRNA in, you cannot continue translation. Chloramphenicol is our next ribosomal targeting antibiotic. This is made by the bacterial genus Streptomyces, but now it's pretty much just produced synthetically in a lab rather than grown up by our actual bacteria. It is bacteriostatic, which means it inhibits growth of bacteria without directly killing them. But the good thing about it is that it is a broad spectrum antibiotic, so we can use it to treat both gram-positives and gram-negatives. Its structure is shown over here just for reference. It's a relatively small molecule as far as antibiotics are concerned. This one works differently from tetracyclines, where instead of binding in the A site, chloramphenicol actually blocks peptide bond formation. So normally a peptide from our A site would be bound to the peptide from our P site, making our little peptide chain growing out the top. Chloramphenicol prevents peptide bond formation, effectively inhibiting translation, because without peptide bond formation, our amino acids cannot be linked together to make a whole protein. The aminoglycosides are one of our most diverse class of antibiotics, still targeting the ribosome. They are made by the genus Streptomyces, and the name of the original one comes from this, streptomycin, but you may have also heard of some of the other ones, kanamycin and gentamicin. Different structures, again, are shown on the right. And you can see they all vary a little bit from each other, but they work in the same way. They are broad spectrum. They can target gram-positives and gram-negatives. One of the drawbacks to the aminoglycosides is that they cannot be used for a prolonged amount of time because it can cause kidney dysfunction and kidney issues. So any patient that's going to be on these for a while needs to have their kidneys checked regularly to make sure that there's not any issues occurring. Aminoglycosides actually bind to the 30S, or the small ribosomal subunit, and in doing so, they prevent proper translocation of the ribosome. So translocation is when the ribosome moves one codon or 3 nucleotides length in order to read and bring in the next tRNA. The binding of aminoglycosides to the 30S subunit prevent it from moving that full codon's length. And what we wind up with is misreading of the messenger RNA. And so rather than making the normal protein, the ribosome makes a faulty protein that cannot carry out the normal cellular function. And the last of the drugs that target our ribosomes that we're going to talk about are the macrolides. Again, made by the genus Streptomyces, these bacteria make a whole lot of different types of antibiotics, and some you may have heard of are erythromycin or azithromycin. Those are the more commonly prescribed of the macrolides. They are bacteriostatic, so they inhibit growth without directly killing. They are broad spectrum, but are particularly useful against gram-positive, and are also particularly useful against some gram-negatives that are harder to kill, like Neisseria species and Legionella. The structure of erythromycin is shown over here on the right. All of the macrolides contain these large lactone rings, Otherwise, their structures coming off of those rings can vary from chemical to chemical. In general, the way the macrolides work, and in this case we're looking specifically at erythromycin, is they bind to the 50S subunit, the large subunit of bacterial ribosomes. And in doing so, they prevent translocation. So they prevent the movement of the ribosome one codon length down the messenger RNA. And again, no translocation means no protein. Next on our tour of antibiotics are those that target nucleic acid synthesis. And we've talked about these before also. The major group are the quinolones. You may have taken something like nalidixic acid at some point. This is it over here. And this is just a base quinolone. You can see they're very, very small compared to our other antibiotics even. And you may have also heard of or taken ciprofloxacin. This is a fluoroquinolone, so it's just slightly different from our regular quinolones as shown over here. Quinolones can be semi-synthetic, so we can take the base quinolone molecule, modify it in the lab to give it different groups coming off like the gray ones that we see on the figure. Or they can be completely synthetic, where they're only synthesized in a lab, we don't even begin with a natural product. These type of antibiotics are really good against extracellular pathogens, so bacteria that grow outside of our cells, but they're not very good against intracellular pathogens. They are, however, broad spectrum, so they're nice and easy to use for a variety of types of infections. Recall that these type of drugs that interfere with nucleic acid synthesis, they don't prevent the polymerase from working, but they target things like DNA gyrase. And again, this is an enzyme that prevents supercoiling of DNA, and they can also bind to DNA topoisomerase 4, which is actually responsible for nicking the DNA. So this one nicks it, and then gyrase twists it to prevent supercoiling. If we inhibit those functions, there's too much supercoiling in the downstream DNA around here, and it will snap and break and the cell will die. And the very last antibiotic that we're going to talk about are ones that inhibit folic acid biosynthesis. Folic acid is a precursor molecule that is needed for the synthesis of purines and pyrimidines. If we inhibit the formation of folic acid, bacteria cannot make their purines and pyrimidines. They can't make all those A, C's, T's, and G's that they need for DNA, and this will kill them. These types of drugs are the sulfonamides or sulfa drugs. Trimethoprim is the most used of these. Here's its structure on this side of the slide. Unlike most other antibiotics, these are completely synthetic, so they've only been generated in a lab. They don't come from any natural source, completely synthetic molecules. And they are, again, bacteriostatic. They will inhibit growth, but they don't directly kill the bacterial cell. But they are still very, very effective at treating certain types of infections. In the synthesis of folic acid, a molecule called PABA, which has this 6-carbon ring here in the middle, gets converted to folic acid by the addition of a couple of side chains that we see over here. The way sulfonamides work is that they share this 6-carbon ring with PABA, and they also have this nitrogenous side group. This 6-carbon ring binds the active site of the enzyme that would convert PABA to folic acid, one of those steps, and when the sulfonamide gets in there, it blocks the rest of folic acid synthesis from occurring. So we're clogging up the active site of the enzyme, and bacteria can no longer make nucleotides. Time for a rapid recap. All right, we have drugs that can target peptidoglycan. Mostly they target transpeptidation, but they can target other parts as well, like the bactroprenol. Antibiotics can also target ribosomes in a variety of ways, and there are a whole lot of antibiotics that do this. Just as important were those that targeted nucleic acid synthesis, preventing DNA replication by inhibiting DNA gyrase and DNA topoisomerases. And last but not least, those that target folic acid synthesis, and thereby prevent the formation of A's, C's, T's, and G's that the bacteria need to replicate DNA and make more of themselves.